Okay, good morning, everyone. Thanks all for coming. So you're probably not as excited as I am, but I'm happy to see you all here. This is the first time this course is being given, Cell Biology by the Numbers. I hope you'll enjoy it. I'm Ron Milo, and the TA of the course, Uri Bernholz, is sitting here. So what will we be doing today? So we'll start by giving some motivation for quantitative cell biology, talking a bit about the structure of the course. Then I'll be moving on to give a case study to try and motivate what are we doing in cell biology and get some hands-on experience on that. And then I'll be talking about what is expected of you throughout the course. So far, so good. Throughout, I will be expecting that you ask me anything that's not clear. Don't wait until the end of the recess. Okay? So let's start by talking about some of the aims, where the question is basically why quantitative biology. And I think this is a fair question because if we're thinking about the condition of biology today, molecular biology, cellular biology, I think it's fair to say that there's been tremendous advances by focusing on qualitative reasoning, on looking at things and interpreting experiments in a way that's mostly qualitative. And yet, what I'd like to show is all sorts of examples of where we could use our quantitative understanding in order to gain more and use that as another tool in our arsenal in order to be able to interpret experiments, to plan experiments. I can take as an example thinking about cancer. So trying to understand cancer, I think we've learned a lot. And the picture that we have today of the complex pathways of signal transduction related to cancer are giving us a lot of insight into what happens there and to trying to move towards cures to that disease. But at the same time, a good friend of mine, Itai Yanai from the Technion, was telling me, you know, usually when talking about cancer, you would hear that somebody got cancer. It would be a very sorry announcement. But you usually think about such things as cancer being sort of like a binary thing. Somebody has cancer, some other people do not have cancer. Where actually the more we understand about it, we understand that cancer is a process that happens to some extent in cells throughout our body, in everybody to some extent. And the question of whether that would lead to what we usually term as the disease of cancer, that's really a quantitative question because it's a sort of a temporal dynamics of how many cells, at what rate, how much it would reach one threshold or another threshold, and what would be the dynamics of those things. And in order to be able to go forward from just knowing the pathways involved in that into being able to project what would happen, to be able to deal with it in different ways, we need to focus also on those quantitative aspects, and that could give us some more insights. So going from that, I'd like to say a few specific things that I hope that as aims would come out of this course. So one thing would be to try and improve our intuition about if you like, what it means to be a cell. So intuition is very useful. And I would like you to be able to come out of this course with a better understanding. Say, if I'm thinking of myself, the size of a bacterial cell, let's say an E. coli, what then would be the size of my other model organisms? If I'm the size of an E. coli, what would be the size of a yeast? That would be roughly an elephant. If I'm E. coli, what would be the size of a human cell? It would be the size of about a big whale, of a blue whale, right? The largest known animal. And in a similar manner, you'll find that in many cases, our day-to-day intuition that has been useful for us in many ways does not work the same way when we think about what happens in the microscopic world. For example, if you're thinking about a bacterial cell moving around, when it doesn't have food, it starts doing locomotion. If you think about the speed at which it moves, so say an E. coli cell using these motors, fantastic motors that it has, it moves them in 100 hertz, really, really fast, and it drives it in 20 microns per second within the media. What happens if it would stop? Our intuition, we have some intuition what happens when something stops. When something stops moving its motor, and it sort of like glide until some position. Actually, when one looks at what would happen at the scale of a cell, after it stops moving its motors, after how long, any guesses, how long would it take until now it completely shuts down and stops? Throw out some guesses just to keep us all entertained. In terms of the, let's say, the distance that it would pass. One cell length. That's a good intuition, I would say. Very often, the answer to things are related to the length scale of the problem. So a length scale here could be the cell length. So that's about a micron. Any other guesses? Intuitions? 20 microns. OK, that's on the order of what it would pass in a second. Actually, it's less than an angstrom. An angstrom, the size, that's 0.1 nanometer. That's about the size of a hydrogen atom. Meaning it would take less than one atom of water, one molecule of water, until it shuts down and stops. That's very different from what would happen to me if I'm going to the pool here on campus and I stop swimming for a second, I'll still be moving ahead. Definitely would happen to me also with my car. But what happens at the microscopic level could be very different. Similarly, if we think about the energetics of a cell, so the cell fuels itself through respiration, through metabolism, we'll be talking about what happens if, for example, it shuts down because we've put some drug on the cell or because its cell resources have dwindled to a level it doesn't have more anything to burn or to use. How long would it take until it uses up its resource of, say, ATP or its reducing power? And we'll find there again that the timescales could be extremely short. And that gives a perspective on what needs to happen in terms of, say, regulation of these processes and things like that. So that's something about our developing our intuition into what happens at the level of the cell. Let me just say one other thing. So in this first lecture, I'm giving quite a few directions, examples. I hope that would serve as some sort of a teaser into what's going to happen next. But I'm also knowing that it's only a teaser in terms of I won't be going very deep in some of the examples, except maybe for the case study that we'll do. Please allow me that freedom, and I hope that in the next few lectures we'll have time to elaborate more on this and on other things. OK, but if you feel uncomfortable because you're not sure that we really got to the bottom of something, it's good. It means that your level of critical thinking is at the right position. OK? Then another thing that's a main aim is to develop our ability to perform sanity checks. And sanity checks refers both to, somebody comes and give a talk, and while the lecture is going on, you want to be able to see if the hypothesis that they're suggesting, whether it really makes sense or not. And we'll find that very often you can do it on the back of the envelope on something that while listening to the lecture. And it's even more useful, I hope, for experiments that you yourself are doing and hypotheses that you yourself are suggesting. So this is definitely one of the main take-home tools that I hope you'll come out from this course. I hope also it would mean that you'll have much fewer boring lectures while listening to seminars, because you can always use the time in order to do all sorts of sanity checks on what you're hearing if you wish. As an example, I was driving to a conference in Jerusalem with a brilliant student from Tzachi Pilpel's lab. Hila Gingold, and she was telling me about an idea that she had during her, when she started studying here, and that was about thinking about the utilization of tRNAs in order to read messages of mRNA. So you know, in the process of translation, the ribosome goes through the mRNA. Every time it charges, you have some charged tRNA that comes in, and then it's being released. And she told me she was very curious about the fact there could be different synonymous ways of having, different codons for the same amino acid. And she said, wouldn't it make a lot of sense for the cell to try and optimize things in a way that it would use the same type of tRNA for adjacent codons that are supposed to give you the same amino acid? Okay, if you have the same amino acid, why not use the same tRNA such that when one of them is incorporating the amino acid, the tRNA is being released, we could reutilize it afterwards, the same one, instead of using something else that would take time to diffuse and whatever. Okay, it's a hypothesis. And I hope that throughout the course, you'll see how we could approach that while driving to the conference to already check, whether, except for it being a qualitatively reasonable statement, whether it could also quantitatively be reasonable for the cell or not, whether that could be achieved or not. Okay? In the end, it's interesting. She started working on other things. There were lots of other interesting projects. Then it turned out there was a Cell paper that was talking about this work. And maybe we'll even get to showing all sorts of caveats with what they did there and what can we learn from doing this type of sanity check. OK, so that was about sanity checks, both of things that you hear from other people and about things that you yourself do. Then I hope as an aim is that this would also serve to suggest questions. And I would offer you to try and ask in many ways why questions about all sorts of properties that you study in biology. So for example, when looking at regulation and control, you often find, say, I'm studying a lot metabolism, you often find that regulation happens first and foremost through what's known as allosteric regulation. Okay, regulation in which, say, the product of the pathway or something related to that or something in the input to the pathway would be regulating the system, regulating the enzyme's activity, etc. But as you know, there are many other ways of regulation. It could be transcriptional regulation, there's translational regulation, there's all sorts of different types of post-translational regulation. And you can ask the question, why use this type of regulation rather than another type of regulation? Okay, some people are quite afraid of asking why type questions in biology. I would suggest that at least in the context of this course, I think that could be very useful for us. In a similar manner, you can ask on anything that you measure, say your favorite protein that you study, if you're measuring its concentration or some metabolites and you know its concentration, I would suggest that you ask yourself, what would happen if the concentration was, say, 10-fold higher or 10-fold lower? Why have the level at which it is? Is there any fitness advantage here? I'm not saying that always you'll have a good answer that it has to be a fitness advantage. Sometimes there's neutral drift. Sometimes the cell doesn't care so much. But I think it's a good exercise to ask these questions in many, many different contexts. OK, just a final example. Many bacterial cells, when they take up, say, glucose from the media, the first thing that they do to it already at the level of the transport is to phosphorylate it. You take a glucose from the outside, you phosphorylate when it's inside. You can ask yourself, why is it necessary to phosphorylate it directly? Or maybe it could wait, maybe it's not even necessary. And actually, I would suggest that there are good quantitative reasonings for that. In the case of glucose, if it's not being phosphorylated, glucose that's not charged, the cell is partially permeable. The glucose, after being taken up, could easily be lost through the membrane. Okay, that's a qualitative statement. But is it really important or not important? For that, one needs to do the calculation to see what would be the time scale for losing the glucose after it has been imported. So we'll try to see how can you do such calculations. Okay, for achieving basically all of those aims, the main tool at our disposal, and something that I hope would be the main thing that you take from this course, would be the skill of estimations. There's more to it than meets the eye. You've probably done it several times. I would suggest that I find for myself that, in preparing for this course, I'm still learning more and more things on how to do it properly. And in a way, it's an experiment for all of us to try and develop our skills in that direction. Anyone's interested, I brought here a collection of all sorts of books that throughout the years I enjoyed seeing examples of how this is being done in other fields of knowledge. For example, it's very common in physics, in engineering, and in environmental studies. And in the field of biology, we're actually trying to develop the methodology of doing that as well. You'll see here a draft of a book that I write together with Professor Rob Phillips from Caltech that's focused on that with the same title of Cell Biology by the Numbers. And I do hope that throughout the course, from your questions, from things that you write to me, you'll get into the acknowledgement sections, because there's still lots of space there for many good ideas that would come out of you suggesting things and helping us make the book better. Okay? So, in thinking about estimations, a person that has been quite influential on doing these things is the physicist Enrico Fermi. And actually, the problems of doing these types of back-of-the-envelope calculations are known in many fields as Fermi problems. So the most famous example so Enrico Fermi, it's being told, used to do it throughout his life. In he would suggest that, students, colleagues would try to attack him with all sorts of questions of trying to estimate something of any type or sort, and he would have a lot of fun trying to calculate that. I would hope that also in this course, in the recess, between lectures, or in real time, you're welcome to suggest questions that you think would be interesting to estimate. The most famous example that is being mentioned in his name is the fact of trying to estimate the number of piano tuners in Chicago. And it's an interesting case study of challenging a person to try and see how, from just without ever being in Chicago and maybe never playing the piano, you can still try to estimate that problem. So we'll be talking about how to perform such estimations. Today, it's actually, even if you won't find that it's useful for your PhD in any way, as it turns out, there's more and more, I hear that people are using it in job interviews. So it seems that Google is very fond of asking a question like, if you have a truck being filled with ping pong balls, how many balls would fit in there? I've heard that one of the consulting companies is asking, if you took the road from Tel Aviv to Jerusalem and you need to paint the separating white dashed line, how many buckets of paint do you need to bring with yourself in order to do that? And if other people have other examples, I'll be happy to hear, but there's all sorts of different fun things you can try and estimate that way. Another way to pass time if you're bored at any point in this lecture or in any other lecture. And you could also, I would hope that you'll find example to use it for your research, but also you can try and bring it to heart and, for example, try to estimate what fraction of the people finishing the PhD at the Weizmann Institute would turn out in the end to have position in academia, say in Israel. That's also something amenable and maybe closer to the heart of what you can estimate with such things. Okay. Anyone before moving to, so next thing we'll do is a case study. Before we move to that, any interesting Fermi problems that people have ever done or heard about? Cards that are in the world or in Israel. Never heard that one. Good.
How much sample buffer do you need to prepare in the beginning of the PhD to estimate?
Excellent. How much sample buffer you should begin if you want to just make it on your first day of PhD, that it would last for forever for all of you PhD. I love it.
How many questions were asked in cell biology by the numbers courses around the world?
Okay, that's an interesting one. So saying how many questions were asked in cell biology by numbers courses throughout the world. That's an interesting one. What?
Including or not including this one.
Okay, good. So we're off to a good start, I hope. And what we'll be doing, our first case study will deal with the following question. How many mRNA molecules are in? And by the way, if somebody already did that specific question with just not long ago, please don't shout the answer, okay? Are in a bacterial, and when I'm saying bacterial, I'll be thinking about E. coli for now, cell. And to make life easier, let me give you some options, okay? We'll make this quiz with options. Option 1 would be 10 to the 3 to 10 to the 4. B would be 10 to the 5, 10 to the 6, 10 to the 7, 10 to the 8, or 10 to the 9, 10 to the 10. Is the question clear? Okay? So we're talking about not species of mRNA, but the total number. You count if one of them has lots of copies, you count all the copies, you sum everything over the whole set of genes. Okay? So if the question is clear, who's in favor of option A? On the order of 10 to the 0 people. How many are for B? Quite a few more. Option C, about the same. And option D? Okay, so we have some normal distribution. Okay, so what I suggest we do, we'll be talking about this problem now. But before that, what I'd like is that people would pair up with somebody sitting next to them. If it's somebody they don't know, that's even better. And let's take a few minutes, let's say 5 minutes, to try and both think about the question and try to devise a way. How would you try, and if you were Enrico Fermi coming into biology, how would you try to perform such an estimation? It's fine if you don't know how to get even to the value. Just try to think, what numbers you would think, or just to build the strategy of trying to approach the problem. Okay? So far, so good. It was a good idea.
Rate of mRNA synthesis, the rate of mRNA degradation, but mainly the number of proteins that the cell needs to be translated.
Okay. יש איזה טקטיקה?
הטקטיקה שלנו הייתה פשוט לחשוב על הניסויים שלנו.
אוקיי. ולמה זה הוביל אתכם?
זה הוביל אותנו לתשובה השנייה, אבל אנחנו עובדים על human cells.
אוקיי. Okay, let's try. I'm happy to see that I could go and come back in half an hour, but let's try and now move through it didactically, steps after step. Okay, so the question is clear. I already know, I've heard from some people there are all sorts of strategies to do it. So what I'll be presenting is one approach into trying doing it. In general, it's always good to have more than one strategy, and that's a good sanity check on the sanity check. Let's start with one.
But you first need to do another vote to see how people...
Okay, yeah. Good. Okay, excellent. So this is the second round, just like in the municipal democratic elections. So who's voting for A? Who's voting for B? Who's voting for C? Who's voting for D? Okay, so there's definitely a change of minds, which means that maybe you can skip the course. You already know how to do it. Everything's fine. So let's start with talking about the basic strategy of how one could approach it. And very often, when looking at these back-of-the-envelope calculations, Fermi problems, trying to estimate how many shopping carts there are in Israel, a main thing, the question itself might sound a bit daunting, but the thing that would help us is to perform divide and conquer. I hope I'm not doing any. So the idea is to break it down into small pieces in which you might have some good estimate, good reasoning, guess from somewhere, and then bring them together so that in the end you'll be able to reach the final answer. Okay? If it was the number of shopping carts in Israel, what would be your divide and conquer strategy? How many supermarkets? Okay, so we had how many supermarkets in Israel, how many shopping carts per supermarket. Okay, so this is the first step. But now still, how many supermarkets in Israel, that might also be a bit tough. Okay, so you can start with how many people are in Israel, and then how many people do we think are required per supermarket, etc., etc. You'll start to break it down, and there are different ways of trying to do it. Okay, let's try and do it in the context of cell biology. Okay, so we'll be starting with cell size of an E. coli. That will enable us relatively easily to get to the cell mass. From that, by the way, we'll have to be doing some unit conversion. That's something that we'll be doing a lot. That we'll have to, when moving into cell mass, we might need to do conversion from one type of unit to another. So we'll probably be talking about Dalton units. We'll get to that. And now we'll need to bring in some other information, and that we'd like to go from that into the protein mass. From protein mass, we'll go to the number of proteins. From number of proteins, if you also have information on the cell cycle, you can know the production rate of proteins. To that, we'd like to add in the protein per mRNA production rate, which would basically give us the number of mRNA per cell, which is what we're looking for. Okay, so that's a suggestion for performing the kind of divide and conquer, or the flow of the logic of trying to approach that. Okay, so far so good. So let's start to move in that direction. So first thing, we want to think about the cell size. So we're talking about bacterial cell, type of E. coli. How does it look like? So, it's usually drawn as this kind of ovoid spherical shape. And what's important to know, very often in biology and other things, everything being drawn the same way. There's a nice book here called Consider a Spherical Cow, because whenever you want to ask questions also in other fields, you're starting with thinking about spherical cows. Excellent book, by the way. And for that, what you usually do is you want to get the reasonable length scale that's relevant to it. So what's a relevant length scale in thinking about bacterial cells? 1 micrometer. Okay, what's a micrometer? 10 to the minus 6 meters. Excellent. And so from that, we'd like to go to the cell size. So in talking about cell size, I'm talking about the volume per cell. So that would be 1 micrometer cubed, 10 to the minus 18 meter cubed. Okay, whenever anything is unclear, just let me know. Okay, moving from that to the cell mass. If this is the volume of the cell and I want to get the mass of the cell, this would be the volume times the density. So I have 10 to the minus 18 meter cubed times, what is the density of the cell? So first of all, it's very easy in biology, because with densities, there's not the kind of huge differences in scales that you would find for other things, right? It's not like it could range from 1 to 10 to the 6 or 10 to the minus 9. It's all around 1, okay? In biology, almost everything sits somewhere between just below 1, say some lipids, etc., which could be a tiny bit lower than 1, to say 1.8 or maybe 2 of the heaviest things that you can find, within DNA, chromatin, grains, other things. Okay, but then what is all this 1 that we're talking about? So 1 is a definition in comparison to water, okay? So how much does water weigh? So water weighs 1 kilogram per liter. And if we're talking here, so this would be 1 kilogram per liter. That's fine. And actually, so by the way, the accurate value for E. coli is about between 1.08 and 1.1. Okay? But as you'll see very often when I'm trying to do, if it's 1.08 or 1, for telling you which of these answers is correct does not matter at all. Okay? So we saw like throughout these calculations, we're keeping in some mental picture of the accuracy level that we're interested in, whether we actually want to know just the order of magnitude, or we want to know it to a factor of 2, or we want to know it to within 10%. It depends on the question. And according to that, we're allowing ourselves the freedom of how to do like convenience calculations of how many digits you want to use. Okay, so 1 is good enough. But then we still have a problem here that this is, the units here seem a bit funny, right? We're mixing here between meter cubed and liters. So in order to do that, we have 10 to the minus 18 meter cubed times 1 kilogram per liter. How would I move such that in the end I just want to have mass, right? That's what I'm looking for. So often you probably did it, but at the same time, it's really easy to get confused. Here, it's not so bad, but you'll see in examples that we'll be doing later. What I definitely suggest is to give yourself the freedom to always multiply by 1, right? Multiplying by 1, there's nothing bad that could be said about it. And by multiplying by 1, you can always get the units right, because what I want to do is I want to convert between what? Between meter cubed and between liters. So what do we know about the connection between them? How many liters are in a meter cubed?
1,000.
1,000. So I have 10 to the 3 liters per meter cubed. Or I could even write it maybe more clearly. As 10 to the 3 liter, 1 meter cubed. I know, by the way, I'm going very slow here. Bear with me, okay? I think it would save you some mistakes of 15 orders of magnitude in your future. It's worth the investment of a few more minutes on that. So 10 to the 3 liters, 1 meter cubed. This is just by definition equal to 1 because that's the unit conversion, so you're allowed to do that. And then that makes life easy because now, in terms of units, meter cubed would fall with meter cubed, liter with liter, and I just need to do the multiplication. 10 to the minus 18, 1, 10 to the 3, so that's 10 to the minus 15. What are the units now? Kilogram. Okay, what is 10 to the minus 15? Is there any? Okay, so it's good to everybody exercise together. We have milli, micro, nano, pico, femto. We'll have to remember at least that. So that's also equal to 1 femto kilogram, which is easier for me to remember as 1,000 femtograms. Okay, here in the last step, I used the fact that there are 1,000 grams in a kilogram. Okay. So we've arrived at the cell mass. Now we have to make a jump. All the things that we want to discuss later, they are in the microscopic regime. Okay? These are proteins, mRNAs, all of those things. They prefer to use different units than the units that we love to use, like these MKS standard units, metric units, that relate to our macroscopic experience. Okay, so we'll have to do the jump from the macroscopic units to the microscopic units. Not a big deal. So what I'd like to do is now to move from the 1,000 femtograms into Daltons. So a reminder about Daltons. So Daltons would be our bridge very often from the macroscopic world to the microscopic world. So a Dalton, by definition, is the mass of hydrogen atom is 1 Dalton, and an Avogadro number, Na, Avogadro number of Daltons is equal to 1 gram. Okay? And if we got that far, anyone remembers by any chance how much is the Avogadro number? 6.0 something, something, something. And it's funny because often, these things, we might remember them or not, but actually the thing to remember is that's almost not important. It's times the 10 to the 23, minus 23. Okay, plus 23. Okay, how can we think about it? Because we need a lot of Daltons, a lot of hydrogen atoms in order to give us 1 gram. Okay, and throughout the way we'll be approaching that, we'll definitely be forgetting about the 6.0 whatever. 6 times 10 to the 23 would serve us very well. Actually, we'll even sometimes allow the freedom to use this 10 to the 24. And whether or not we're allowed to do that, that depends again on the level of accuracy that we're looking for. Whether we find we just, so how much of a mistake did I do here? Somewhat less than twofold, right? So it depends. If I just want to know orders of magnitude, maybe I could allow myself that freedom. Okay. So with that, let me see, what do we want to erase first? Let's finish there, and then we move to here. So we have the mass of the cell, which is 10 to the, it's 1,000, excuse me, we'll write it that way. 10 to the minus 15 kilogram, which, because I want to use the Avogadro number, which works with a gram, I can also write as 10 to the minus 12 grams. Now I want to move to Daltons, and I'll do it again with the same thing of multiplying by 1, which I'm allowed to do, and that is that there are 6 times 10 to the 23 Daltons per 1 gram. Okay, so what do I have here? The gram and the gram units would fall off, and I'll actually have the Daltons, which I was looking forward to having. And now I have 6 times 10 to the minus 12 plus 23. So that's 11 Dalton. Okay, so that's the mass of the cell in the microscopic units of Daltons. Okay, so we got so far. Now we want to go to the protein. We want to incorporate protein mass in order to give us the number of proteins. And we'll be doing it so out of that mass of the cell, I want to move to the mass of protein per cell. So should I take the whole number? I should only take a fraction of it. So what do we have in cells? What's the main component in terms of mass?
Water.
Okay, so the main component of this would be water. How much roughly? So 70% or 2/3 would be a good rule of thumb on that. Okay, out of the rest, what do we have? We have membranes, lipids, DNA, RNA, sugars, proteins, right? Proteins are we? Okay, so there are different things. And in the third lecture of the course, where we'll be talking about composition and concentrations, we'll be playing around a lot with these things and have a lot of fun with it. For now, I'll just be saying that proteins, say for bacterial cells, for not, except for some very specialized cells, in our body or in plants, etc., proteins are usually the dominant factor in the cell dry mass and are about half of the cell dry weight, CDW, so half of the remaining. Okay? So that means that the mass of protein in the cell is the mass of the cell times about 0.15, 15%. That's because 30% is the cell dry weight. Half of it is protein, so that's roughly that. So what do I have? I have 6 times 10 to the 11 Dalton times 0.15. And that's about what? So that's about 10 to the 11. Okay, you can see that I'm not being super careful. 6 times 0.15, that's closer to 9. I don't care about 9 or 10. or excuse me, that's 0.9. I could make it a 1. So I have 10 to the 11 Dalton. That's the mass of the proteins inside the cell. OK, we're getting closer. Now, in order to move forward, I'd like to know the characteristic mass of a protein. So how can you help me with that on the mass of a protein? So we could do it in different ways. Anyone knows anything about the length of a protein? 300 amino acids or something like that. OK, that's again, and how much is the mass of an amino acid? 110. OK, so one way to do it is to say, when I'm looking at the mass of the protein, I'm saying there's about 300 amino acids. 300 amino acids. I multiply it by about 100 Dalton per amino acid, which gives me 30 kilodalton. OK, 3 times 10 to the 4. And that actually, I think, as a sanity check, I've already heard somebody from the crowd before saying it's a few tens of kilodaltons. OK, so that's about right. So from that, we could get the number of protein per cell by doing what? Just dividing the mass of the protein in the cell by the mass of a characteristic single protein. So that's 10 to the 11 Dalton. I should have written it. That's just a nice way to remember, but that was 3 times 10 to the 4 Dalton. And I'm getting what? 3 times 10 to the 6. And I'm writing here, this is proteins per bacterial cell. So we're still on our way to answering the quiz, but I already argue that this is that might actually be very often a more interesting number than the quiz that we started with, because proteins are such a dominant fraction of the composition of a cell and something that people love to study throughout. It's good to know something about the number of proteins inside the cell. We'll be talking later also if we want to look what happens in different types of cells. Also, I would say we should take this number with a grain of salt, because if we're thinking about what came into the calculation, what could go wrong? Okay? Let's say I told you somebody wants, looking at this number says something is fishy, it's not exactly what I'm getting. What could have gone wrong in what we did so far? What would be the strongest suspect in the parts that we put in? Yeah. Right. So an option. So you're saying this 300 amino acid, that's just an average enzyme. Maybe it's wrong by somewhat. Okay. Other suggestions?
15%.
What? 15%. 15%. So you're saying we said 15% of the protein of the cell mass is proteins. Maybe it's different. Okay. Cell size. So we used 1 micrometer, or actually we used 1 micrometer cubed. So okay, so I would say this is good in terms of this sort of analysis known as sensitivity analysis. When you have some model and you want to see what are you dependent on and getting the final result that could actually shift the value of it. It's very useful in engineering and doing models of all sorts of different things. In this case, my intuition would say that that definitely could be wrong, but I would be surprised if it's wrong by even. It would be hard for me to think it's more than twofold, right? Instead of 300, it might be 250 or maybe even 400. Similarly, 15% here, given that you need to have a lot of water, etc., it might be 0.2, maybe 0.1. With the volume of the cell, cubed, there you can really go wrong. Because actually, when you look at sizes, at cells like E. coli, their cell volume could vary by quite a lot depending on the growth conditions. So when there's excellent growth conditions, when they're getting this heavenly medium of LB with all of the different components and a wonderful temperature, they'll be growing really, really fast. They could have a volume that's actually something like 5-fold or even more larger than what happens when they're getting some crappy media that's maybe even now they're getting into stationary phase because they're finished all of their carbon or sugar within the media. So this is a place where you could have quite a lot of variability. So 10 to the minus 6, that's a good characteristic size, or 1 micron cube, that's a good characteristic size for bacterial cell, but it could vary several-fold, and that would definitely affect our result here. In that respect, I would suggest that a good way to interpret this result would be to say this is 10 to the 6, 3 times 10 to the 6 proteins, not per proteins per bacterial cell, but this is actually 3 times 10 to the 6 proteins per micrometer cubed. Okay? This would be a much more robust statement of what we got as a result than just talking about the cell could vary by a lot. But when you do that, then you're much better in terms of the accuracy you can expect. That would also make it much easier for you to extrapolate for other conditions. So many people do not care about bacterial cells, and that's completely fine. If you want to think about a yeast cell or about a human cell or about something else, if you have this at your disposal, you can easily try and say something if you know the volume of the cell. That's assuming that you feel comfortable about the things that we incorporated here, the number of amino acids per protein and the fraction of protein within the cell. And we could talk about it, how much inaccuracy that brings in, but I would say that's a good rule of thumb to carry with you. I mentioned about rule of thumb. You'll see that we're using several of them throughout. It might look a bit confusing because, it's like a magician. Where does he grab these numbers? So first of all, you see that I'm using the crowdsourcing in order to get the answers. So actually, some people carry those with them. And as you'll see by the end of the course, I think you'll find that there's actually a very small subset of numbers that you need to know. And with them, you can do a lot. So I hope you'll find them useful. And we'll also be giving you a list of key numbers that from the lectures that you can carry with you. And once you have them in store, you can do all of those calculations. OK, so let's move on. We have the number of proteins. So per cell, we got so far. Now I'd like to try and finish up. And for that, I'd like to know the rate of protein synthesis. And for doing that, we'll have to know something more, and that is the cell cycle. So what can you tell me about the cell cycle? What would be a reasonable value for the cell cycle? Half an hour, 20 minutes, sold. OK, so 20 minutes would be really, really the fastest one could expect from a bacterial cell. And then, by the way, the volume would be much larger. And on the upper side, it could be really, really long because they could be in stationary phase. So in terms of using a value for the cell cycle, so often denoted by tau. I'll be happy to use 3,000 seconds or 1 hour. That's all some characteristic value of a doubling time, and also that would be quite useful in a second in terms of the numbers that we already have. Because the rate of protein synthesis would be what? Would be the number of proteins per cell divided by tau. So we have three times 10 to the 6 proteins per cell divided by 3 times 10 to the 3 seconds, which gives us 10 to the 3 proteins per second per cell. Okay, so the cell needs to, a bacterial cell needs to produce about 1,000 proteins per second. Yes?
I don't know, I kind of feel uncomfortable with that.
What part of it?
You're assuming so that each cell makes 1 million proteins, and only 1 million proteins, and that's why you do the division, pretty much?
Okay, right. So it's good that you mentioned that. Let me see if I understood why you are uncomfortable. We were estimating roughly the number of proteins inside the cell, and now I'm dividing it throughout the cell cycle. What I was tacitly assuming here is to say that within a cell cycle, the cell roughly doubles itself in terms of volume, in terms of composition, in terms of content. And in that sense, if the number of proteins that I started with was 3 times 10 to the 6, I'll be needing to end with 6 times 10 to the 6, which means that I needed to synthesize 3 times 10 to the 6, which is what I was using here. So this is, again, not accurate to within how much? Roughly a factor of 2, because it could be that this value that we calculated was actually for the middle of the cell cycle. There could be a turnover of the proteins, so some of them degrade and then reproduce. So that's definitely could happen. What is actually known is that for most proteins within, say, bacterial cells, the degradation time is on the order of much longer than 1 hour. Okay, so there it's not an issue, but it's good that you raised that. We'll actually be returning to that in just a second for other things, so remind me to think about it again soon. Okay, so if we have the rate of protein synthesis, so that's maybe another interesting number that we came by. But in order to actually try and reach the final answer, I'd like to now know something about this part. So we got so far as having the production rate of proteins. And now I'd like to know what's the protein per mRNA production rate. And we'll be doing that here. So I want to know the rate of protein per mRNA. So how do we get proteins for mRNAs? What needs to happen? Yeah.
Can we go back to what we did before? In the beginning, we said we have to have around 3 to the times 10 to the power 6.
Yeah, 3 million proteins per cell.
And then you said it takes around 3,000 seconds.
Yeah, so we're assuming that that happens throughout the cell cycle because it needs to double itself. So if it has 3,000 seconds to make 3 million proteins, it would require it to make about 1,000 proteins per second. And now we'd like to connect it to the rate at which proteins are made from an mRNA. Okay, that answers your question? Okay, so we have an mRNA. And on this mRNA, we'll have ribosomes that would be moving around and translating it into nascent proteins. Okay? In thinking about the rate at which this would synthesize new proteins, we can do the following. Think about the mRNA. You have ribosomes sitting on it. And now there's different ways to estimate what would be the rate of synthesis. Let me show you one way to approach it. I'm thinking about the ribosome. The ribosome is moving on the mRNA, churning out the protein. I'm interested in knowing the rate and the velocity at which it moves doing translation. Anyone knows roughly what would be the rate of that? The speed. How many amino acids are being translated?
10 amino acids.
So 5, 10, something on the order of like 10 amino acids per second. That's the rate at which the ribosome would be churning, which means that this is equivalent to about 30 nucleotides per second, right? Okay, now let's think about how many proteins could be synthesized in a unit time from an mRNA. The maximum that could happen, so I'm now getting a maximal estimate, would be if just the ribosomes would be sitting just next to each other. Okay, the messenger RNA would be just filled with ribosomes sitting almost touching each other, each one moving one after the other. And that would give me the maximal rate at which one could synthesize new proteins. So for knowing that, I now need to think about what's the distance that would be among them. That's related, so the distance that would be between the ribosomes, if they're almost touching, that's related to the footprint that the ribosome takes on the mRNA. Okay, so now we're talking about an issue of size. Any intuition about a ribosome, this lovely machine that Ada or not helped self-solve, sitting on an mRNA, what would be the footprint on it? So I hear 10, 15, 20, 30. So I could make 30 nucleotides, roughly for the size of the footprint, say, together with how much can you dense together those ribosomes. This would be if they're touching. So this would now give me the maximal estimation on the protein per mRNA would be if the size is 30 nucleotides and the rate is about 30 nucleotides per second, I would get a maximal estimate of about one protein per mRNA per second. And look, I did an interesting thing here. Instead of thinking, about the length of my gene, the length of the mRNA or the protein, and then how long will it take one ribosome to go the whole way, but then there's a few of them scattered, and then I have to put all sorts of numbers there, and then they cancel out. I'm just thinking, so like at the cashier, at the end of the tunnel, the rate at which ribosomes finish the process of doing the translation, if they're set just like, in some traffic jam, just one after the other, moving all together, that would give me an estimate of how many proteins are being synthesized per mRNA. This is an upper limit. And we could look either, at numbers from the literature or something else. This is like a maximal value. It could range from, say, this, if it's not a maximal value, say, to 0.1 protein, let's say an order of magnitude less mRNA per second. Well, so now I'm doing estimation, or there's a book here called guesstimation. I'm saying the maximal that could achieve is about 1 per second. And let's say it's more dispersed, the cell does not fill with ribosomes thoroughly the mRNAs. It could be, say, an order of magnitude less. My intuition might be wrong, and actually maybe I'm completely wrong, and it's 2 or 3 orders of magnitude away, and that would mean that I'm just wrong. And then the value, so here I'm using my intuition based on some knowledge on what is known on, there's these things called polysomes, which count things on what are the distances of ribosomes on the mRNA. Okay, and with that, we're basically done because I can combine these two things together. The number of mRNA per cell is what? It's the rate of protein synthesis divided by the rate of protein synthesis per mRNA, which is 10 to the 3 proteins per second per cell, divided by, so now let me start from the maximal value, 1 protein per mRNA per second. So proteins falls with proteins, and I have second with second, and I have 10 to the 3 mRNA per cell. This is when I took the larger value. If I'm taking a lower value, 0.1, I'll get, so this would range to 10 to the 4. mRNA per cell. Okay? And now we could make the vote again. Who's voting for A? So along the way, we got an estimation. So you see the main emphasis should be on how we basically decompose the problem into smaller parts and how we moved to get the final answer. We could do it in other ways as well, and that could serve as a good sanity check. Along the way, we needed some numbers that, we should grab either from our intuition or from prior knowledge. And I think throughout the course, you'll get more commonality with how these things can be achieved. I want to say, how can I, just in terms of trying to compare it now to what is known from the literature. So we got an estimate. What do we know from people that actually went and measured it? So it's not so easy to find values on those things, but the values that I could find range from studies that were done already in the 80s. So you can see, for example, from the book of Neidhart, a classical book. So they mention about 1,400 mRNA per cell. A recent study from Germany was looking at what happens in LB media, and they were mentioning a value of 7,800 mRNA per cell. And on M9, they were mentioning something between 2,000 and 3,000. So of course, the value depends on the growth conditions, but you can see that finally in comparing to what is known from the literature, we're at the right ballpark. It might sound a bit, by the way, I'll mention here that there's something that I didn't discuss at all, and that is what happens in terms of operons versus mRNAs, which are not polycistronic, okay? So in bacteria, very often you have a few of them sitting on the same mRNA. I would say that for the order of magnitude, that's not a big deal in a way, because on the average, you have, say, two genes sitting on an mRNA. So it's a factor of 2 that we could discuss how it fits into here, but the order of magnitude would remain the same. In terms of your intuition, there's something that might sound a bit confusing. We got to just this amount, so that means that on the average, I could have, for an average gene, how many mRNAs would I have? Very few, right? Let's say 1. For those that are needed very highly, there'll be more, but then it means that there'll be quite a few that you might have 0 on the average. How does that work out? Right, so this was something that was mentioned here previously about proteins. Proteins are actually quite stable in comparison to the cell cycle. mRNAs are not so stable in comparison to the cell cycle. The turnover rate of an mRNA in a bacterial cell is on the order of 3 minutes. Okay, so if I have an hour cell cycle, it means that I'm on the average replacing my pool of mRNAs how many times? If I have 60 minutes altogether and they're replacing every 3 minutes, I have doing 20 cycles of replacing my pool. That means that effectively throughout the cell cycle, the cell sees and uses a much larger number of mRNAs throughout. Okay, to finish up on this example, I want to say what should we do if we're interested in something else, and not in the mRNAs in the bacterial cell, but in mRNA of a cell that you're of a different cell type. So if I already have the number, let's say I want to take the number of mRNA in a human cell. How do I get at it? So what I suggest is that you can repeat the whole estimate, but another thing you can do is look at how it's supposed to scale. So we already calculated the number of mRNA per bacterial cell. And then I would ask, what would be the main components in moving from a bacterial cell to a human that would affect our result? I hear cell size.
Cell cycle?
Cell size and cell cycle. Exactly. These would be the two components that would be very different and relevant to this. And how would they affect it? So I would need to use here the volume of a human cell divided by the volume of a bacterial cell. And I would need to divide by the doubling time of a human cell divided by the time of the bacterial cell. So if I'm thinking about the specific case of a human cell, you remember we said if we're looking at a normal mammalian cell line, that would be, we said, the size of a blue whale. Okay, the blue whale is very nice, but what does it mean in terms of, say, a volume like micron cubed, some units like that? Any guesses on that? How many people here are studying, in their lab, people are studying mammalian cells, cell lines? Okay, good fraction. So what can you tell us about the size of those cells? Right, so 10 micron, 20 micron would be like a diameter. So let's say if it was 10 micron cubed would be 1,000, right? Okay, excellent. So the ratio of these two numbers would be on the order of 10 to the 3. Okay, what can we say about this ratio? So what is the doubling time of the cell lines that you study in your labs? Something like a day, right? So if we took here 1 hour, this is a day. So this is, say, 20. So if we had here, we had what, between 10 to the 3 and 10 to the 4, right? So now there's a question, if you have 10 to the 3 and 10 to the 4, what value do you take there? Let's put in something in the middle. And we'll be talking maybe in the next lecture of how do you handle these things in an elegant way. It's very often good to take the geometric average, but we could talk about it some other time. So we had 3 times 10 to the 3 times 10 to the 3 divided by 20. So that's what? Sorry. So I have about, this is divided by 10. So I have 10 to the 5 mRNA per human cell. Okay, and you can do a similar scaling, say, for a yeast cell or whatever cell type you're interested in. Actually, when talking about human cells, it could be also a very large variability. We talked about it in the case of a bacterial cell. In human cells, it could even vary much more. We'll be talking about that in the next lecture, probably. Okay, this was our case study, and what I'd like to do in the last few minutes that we have is, first of all, I did want to mention one other thing. So we calculated the number of proteins. We calculated the number of mRNAs per cell. Now you can see that also it could be quite straightforward from here to get all sorts of other things inside the cell. For example, if you already know the mRNA and you were assuming something on the ribosome sitting on it, you can estimate something about, say, the number of ribosomes inside the cell. And you can go ahead, and if you're interested to know for a specific, say you want to do RNA-seq, and somebody told you that you need some mass of mRNA for that, you can now estimate from the number per cell, you can estimate how many cells do you need to take into your experiment. But we'll be talking about such things later on. This is just like showing what's the relevance of these things. Okay, so I'd like to finish up with telling you a few things about what's expected of you and basically the methodology of the course. So because I want you all to come out from this course being able to do these sort of things in your own research and in your life, this is a very much hands-on course, both in terms of the interaction that I hope would develop in the lectures themselves, but also in the things that you'll be doing as exercises. It doesn't, although we'll be talking a lot about diffusion, this knowledge does not come by diffusion or just by me showing it to you. You'll have to play around and try to do it. So we'll be having you both read things and try to do such estimations yourself. The course in total would be 7 meetings. So it's like half a course in terms of the semester, in terms of the numbers of lectures. That would mean that in some cases we won't have it. So we have a lecture next week, but then the lecture after it would be 2 weeks afterwards, and I'll be telling you exactly when and where. Not where. Where is here? Sorry. For anything that you want to check about the time scale and the shape and what is expected of you, you can go to the website of the course. So that's the Weizmann website /CBN for Cell Biology by the Numbers. The first exercise is already there. It talks about estimating how many bacterial cells are in your body. So I hope you'll enjoy that and come up with something that's fun to play with. And please send the answers. No need to print them. We're already printing way too much paper throughout the world, and you can estimate how much if you want to do it. But just email it to cellbiologybythenumbers@gmail.com. Also, if you have any questions, problem, etc., you can email us into that address, and we'll be in touch. I won't have a specific hour every week, but if you just want to meet me, no problem. Just email me, and we'll set a time that's convenient for both of us. I expect you to come to the lectures. There is an expectation to be attending at least 80% of the lectures. I'm already telling you I won't be checking it in any clear way, but I think that the type of course that we're doing here is really based on this interaction, and this is what you need in order to really get out of here from something useful. In the course, there wouldn't be a test. The hope is that throughout the semester and the lectures, you'll be learning and experiencing throughout, and not just studying a few days before and then forgetting everything. You'll be expected to do 3 things. One would be whenever there are specific exercises for the next lecture to do them. So in this case, you'll see on the website, one thing is to do this estimation and to read a paper on that relates to cell biology by the numbers from PNAS that I hope you'll enjoy, and see some historical examples of how that was useful for different things. The second thing for the final grading would be asking you to come up with some creative way in order to challenge from things that you're interested in with quantitative questions in cell biology. So for example, I would hope that you'll come up with questions kind of these that relates to things that you're interested in, and either throughout the lectures or at the end of the course, which will be before the end of the semester, come and present it to the class and ask those questions. So whenever you have some interesting ideas of these, just send them to me through the email of the website and through the email of the course, and you'll be presenting that. Alternatively, instead of coming up with a few questions like this, you can come up with an interesting calculation that maybe you came up with. And that would be also a way for you to experiment with doing these things. Finally, the last thing that you'll be expected to do in the course is to choose a subject. You'll be seeing that from the things that we'll give you to read, there'll be all sorts of small vignettes, which are basically small questions, just like this one, where we'll be trying to give you some insight into why does it matter. Like, what's the concentration of an ATP inside the cell? How many neurons are in our brain? What is the rate of the ribosome doing translation? What is the size of a vacuole inside the cell? So basically, the book on cell biology by the numbers is composed of like 90 such vignettes. So the final exercise that you'll be expected to do is to write a vignette of this type yourself. They're usually like 2-page long or something like that, where basically you need to think about the question that interests you, and then find in your own research or from things that you find in the literature some relevant numbers and something interesting that you can gain from knowing those numbers, some insightful thing. Okay? And I'm quite open to different creative ways of approaching this. If alternatively you want to write a vignette on a number that you yourself are measuring inside the lab, that would be delightful as well. So if you have such ideas, do come to me. A description of what is expected is also given in the website. Let me see. Okay, I would say that this, I'm aware of it, is a course that would require you to do work throughout the semester. As I said, it's not something that you just go before the test and do a crash course of 2 days with somebody that wrote something throughout the course and then go for the test. There's no test. You'll have to work a bit hard throughout in terms of doing some reading and doing some calculations. What I can say is that I hope you come with a reward of being able to do these things, and that would help you throughout in the future. Any questions, things that I forgot? Yes?
Just a semi-technical, maybe a little annoying question. Some of the numbers you gave throughout the proof, some we knew, some I didn't know. In the assignments, are you giving us the numbers that are relevant? Or are we supposed to go looking for them? Or?
So I think for the first exercise, you'll have the numbers that you need, or you've been given them here. Later on, we'll also give you like a cheat sheet, if you like, of basically the numbers that we think are most useful for doing such calculations. And if I didn't supply them in any way, that's also very fair, because this is the sort of thing that you find when you go, into nature, into the world, into academia. So I think you'll have a fair chance of solving all of the problems with what you'll find available, on the internet, in other ways. Some other things, questions, worries? Yes?
Did you mention how many meetings we will have in the course?
Yeah, so we'll have, throughout the course, we'll have 7 frontal lectures like this. In some of the cases where I'm away, we'll also have a meeting with some extra things or explanations given by Uri for those interested. So we'll give information on that as we're getting close. And as part of showing your examples of how you suggested some questions or derivations of doing back-of-the-envelope calculations, we'll be doing that after finishing the 7 lectures, and those that do them will come to present them here in class. More questions?
Also about the email and registration.
Okay, so registration was already open. I know there's quite a few that registered. If anyone wants to take it, not for credit, that's completely fair. I would just suggest that if you want to if we'll be sending maybe sometimes email and updating things beyond what you have in the website, just send us an email to this address and we'll add you to the mailing list of the course. Okay, thanks for coming. See you next week.